Ever wondered why understanding gene regulation is crucial in the field of biology? Let's break it down. Gene regulation is akin to a sophisticated biological switchboard, controlling the development, growth, and functions of all organisms. It dictates when and how genes are turned on or off, influencing every aspect of life. This intricate process is what makes a butterfly different from a bumblebee, or a rose different from a rhododendron. In essence, gene regulation is the blueprint of life. One of the most significant mechanisms of gene regulation is RNA interference. But what exactly is this process? Well, RNA interference, often abbreviated as RNAi, is a natural biological process that occurs within our cells. It involves RNA molecules, which are relatives of DNA. These RNA molecules play a crucial role in inhibiting gene expression. This means that they can essentially turn off or silence specific genes. Now, this silencing doesn't happen haphazardly. It's an orchestrated process that occurs after transcription, thus earning it the name post-transcriptional gene silencing. The process is a key part of gene regulation, meaning it helps control which genes are active at any given time. This regulation can influence our development, health, and biological responses to the environment. So RNA interference, in simple terms, is nature's own way of controlling what genes get expressed. The process of RNA interference is a fascinating journey. Imagine a bustling city at rush hour. Now replace that city with a cell, and the people with molecules. In this bustling cellular city, one of the most intriguing processes is RNA interference, a molecular dance that regulates gene expression. The dance begins with an outsider, a double-stranded RNA molecule entering the cell. This foreign molecule doesn't go unnoticed. It's spotted by an enzyme known as dicer, the cell's vigilant security guard. Dicer, as its name implies, cleaves the double-stranded RNA into smaller fragments known as small interfering RNAs or siRNAs. But the journey of these siRNAs is far from over. They then associate with a protein complex known as RNAi, the RNA-induced silencing complex. Think of RNAi as the cell's detective agency, using the siRNA as a guide to find its target. The siRNA, having come from the double-stranded RNA, carries a sequence that matches the sequence of a specific messenger RNA, or mRNA, in the cell. This mRNA is the blueprint for a protein. It's like a wanted poster, and the siRNA is the tip that helps RNAi find its target. Once the RNAi complex identifies the mRNA that matches the siRNA sequence, it doesn't just mark it, it degrades it, effectively silencing the gene that mRNA was supposed to express. This is the equivalent of the detective not just finding the culprit, but also ensuring they can't cause any more trouble. This entire process, from the introduction of the double-stranded RNA to the degradation of the mRNA, is a tightly choreographed molecular ballet, with each step playing a crucial role in regulating gene expression. It's a wonder of nature that happens countless times every day, in every cell of every organism. And there you have it, the process of RNA interference, a remarkable dance of molecules that helps regulate gene expression. RNA interference is more than just a process, it's a vital part of gene regulation. As we've explored, RNA interference, or RNAi, is an incredible mechanism that cells use to control gene expression. It's like nature's very own volume knob, turning up or down the levels of various proteins in our bodies. This isn't just a fascinating biological process, it's a critical one. RNAi helps maintain the delicate balance of proteins, too much or too little of which can lead to disease. It's not an exaggeration to say that without RNA interference, we would not exist as we do. But it's not only about understanding our biology. RNAi holds enormous potential for practical applications, particularly in medicine and biotechnology. Imagine being able to dial down the expression of a disease-causing gene or boost the levels of a beneficial one. That's the promise of RNAi-based therapies. Already, researchers are harnessing RNAi to develop treatments for a range of conditions, from inherited disorders to cancers. But we're just scratching the surface. The more we learn about RNA interference, the more possibilities we uncover. RNAi also has potential in biotechnology, where it could be used to improve crop resistance to pests or diseases, or to engineer bacteria to produce useful chemicals. The possibilities are as exciting as they are vast. RNA interference, a small process on a molecular level with a massive impact on life as we know it.